We are live. Thank you so much for joining me, Dr. Robert Malone. For those who are listening and watching, thank you for joining us as well. I want to do a bit of an intro. So Dr. Robert Malone is the inventor of the mRNA vaccine technology in the late 80s. He has done extensive research and development in areas of preclinical discovery, clinical trials, vaccine, gene therapy, biodefense, and immunology. Over the last 20 years, he's been involved in leadership and management with the pharmaceutical and biotech industry, as well as you have lots of relationships through government and non-government organizations in the scientific and medical realm, correct?
Correct, yes ma'am.
Okay, so that sounds amazing. For people like myself listening, can you really tell us why you're uniquely set up to speak to some of the topics we'll be touching on today?
Okay, I'm going to start off by objecting to that. I'm sorry, I don't think I'm that unique. Okay. What makes me a little unique, so I have many colleagues that have extensive experience, not many that understand the government, understand industry, understand the science, understand advanced development, project management, regulatory affairs, all that stuff. And it's kind of a consequence of some weird twists and turns in my own career. Gently put, there's the phrase a straight arrow. I'm not a straight arrow, I'm a bent arrow. I've had some transitions in my career. It turns out that the kind of random walk that my career represents, I don't know what things are like for you or your listeners, but I think most of us these days have to be a little flexible and adaptable because the world is changing like crazy. And so this has been the case through my career as a kind of a tail of the dog boomer. But I have millennial children, so I try to be able to communicate with them too. Okay, so once upon a time, I came out of a laboratory at UC Davis that was right at the forefront because of just a cascade of events of learning what the basis was of AIDS as a disease starting in 1983. UC Davis is right next to San Francisco, and of course that was one of the epicenters. And my mentor, Murray Gardner, together with Preston Marks and some other people, Murray he had founded the Cancer Center at University of Southern California before he became the chair of pathology at Davis. And these folks were working at the Primate Research Center in Davis and recognized that some of the monkeys had an immunodeficiency syndrome and hunted that down and found that, in fact, that was a retrovirus that was causing that. And that was before we had really identified the AIDS virus. So just because of how things happened, I was there working on a retrovirus causing breast cancer in mice, mouse memory tumor virus. And I kind of had a front row seat to some very big people and interactions and stuff that happened, much of which was captured in the HBO series film called And the Band Played On. So that's how I started. That was my first outbreak. And these outbreaks are kind of adrenaline rushes. And I've been involved in too many of them now, up until the present. As I became a MD PhD student, I started off a carpenter and a farmhand before all this, but I worked my way through academe, and I decided I was passionate about gene therapy, and so I was young and ambitious, and I wanted to work at the best gene therapy labs. And the best of the best at the time in the late 80s were at San Diego, UC San Diego, in the Salk Institute. And so I went there for my graduate work. And there were two leading gene therapists there, Ted Friedman, who was the person that first came up with the idea, and Inder Verma, who was at the Salk Institute. The Salk being basically the temple that Jonas Salk could build with March of Dimes money. Okay, so really the epicenter of vaccines initially. At the time when I was there was about half a dozen Nobel laureates, including Francis Crick, just to set the stage. Truly, the shoulders of giants, surrounded by the greats of molecular biology and virology. Unfortunately, Ted Friedman didn't want to take me into his lab unless I wanted to do sequencing of whole chromosomes, which I thought was absolutely inane. I'd already done enough sequencing. And if I'd taken his offer, I probably would have been a big shot in the Human Genome Project. Silly me. I wanted to do gene therapy. And so I worked for this other guy, Inderverma, at the Salk in the molecular biology and virology labs there. And I wanted to ask questions about retroviruses that required that I have an ability to put RNA into cells. And that's what started all this, was me building the technology necessary to do my graduate thesis, working in a gene therapy lab that was a pressure cooker. All postdocs, I was the only graduate student, so I was the little kid in the corner. One thing led to another. We could go into the story, but the output was these unanticipated discoveries that gene therapy could be used for vaccines. And the kind of interesting sidebar there is the senior postdoc who was working in adenoviral vectors went on to found the company Crucell. And Crucell was the company that was sold to J&J that produces the J&J vaccine. So it all kind of comes back to that same place. So I was in this discovery environment and made these discoveries in the late 80s, and many patents were filed, and people started fighting over who gets the rights and whose name goes on the patent. And I was kind of caught in the crossfire, and that's a whole nother story. But that's how my career started. Then I went through, finished my MD, became academic, and was very involved in academic discovery research for many years, both at UC Davis and then at University of Maryland, Baltimore, and made a number of other discoveries, helped found the company called Inovio, which is using pulsed electrical fields for gene therapy and vaccines, and other stuff. So I had a career as an academic, high-profile thought leader, but I was always on soft money, and I lost some key grant funding, and I had to scramble because I had to, keep my family alive and pay my mortgage, just like you. So somebody opened a door for me to start working at the company that the Department of Defense had contracted with to develop all biodefense vaccines and biodefense products for the Department of Defense. So I left academia, became an assistant clinical director there, and that was the first time that I really encountered what it takes to make a drug or a vaccine, all the stuff that goes into it. And that was kind of a hard thing. It was like hitting a stone wall. Everything, I had been in this world where we could all think high thoughts and produce our academic papers and, make claims and file patents and all that kind of stuff. And then, bam, I had to be involved in actually making a product. And it is a very different world. The discovery research world is, the Wild West. You can do all kinds of stuff and have all kinds of ideas. Once you get into advanced development, it becomes really structured. And everything has to be quality controlled and project managed and compliant with the FDA, and it goes on and on and on. So I was doing this, and this was right after the anthrax attack, so it was kind of important. And we were working with smallpox vaccines and many other things. It was one of those moments in history. And I had the epiphany that the world really doesn't need more academics. We've got plenty of academic thought leaders. What was really needed was somebody who understood that world, but also understood advanced development and regulatory affairs and all that stuff. And so I kind of said, this is a career change. I'm going to become that person that understands all that stuff, plus all the other stuff that I used to do. And and you can appreciate I was working for the government under contract, and I've kind of kept doing that ever since. And I became, over time, that person. I had, sought out opportunities for learning. I took a fellowship at Harvard for global clinical scholars training, etc., and kind of set myself up to become that person. There aren't very many people that understand that whole spectrum and then have the CV that has all of that earlier discovery stuff that's kind of high profile. And what that does is it makes it so that scientists feel comfortable talking to me about things, because I'm one of them. We can talk sciencey talk, and they know that I'm not. I had somebody tell me once, I heard about you and I thought you just must be another DC hack. And then I looked at your CV. So the CV kind of opens doors for me and gives, lets me talk to people as peers that might otherwise not be comfortable. But I also know all the govies and how all that intricate stuff works. And I've, dealt with people on the Hill and all that. So I know the government pretty well. And I used to live down in Atlanta, and I used to go to the CDC ACIP meetings all the time. So I know that world well. So just one of these sets of circumstances where I've been in these places over time, and I kind of know how things work pretty good. Have I answered your question?
Yes, you have a perspective in, more than one world. And of course, this leads us up to the present day. And you've been speaking out a bit and talking about some concerns that you have and backing up doctors like Dr. Byron Bridle. So this is just a question to get us into things. What do you think are the 3 most important things that the average person should be aware of right now?
Particularly from a Canadian perspective, I'm assuming.
Yes.
And
sure.
I want to say something. Another part of my life is I breed horses. I used to breed Percheron. We still have her, the mare out in the back pasture. As a filly, took first place at the Royal Canadian Winter Fair. Yay!
Yay!
So we really, my wife and I love to travel to Canada and interact with the Canadian people. She's British by birth. She has a British passport and an American passport. And we really enjoy the kind of grounded, forthright honesty that is typical in lack of ego. When I deal with Canadian people, and I think Dr. Bridle was from my interactions with him, he seems to be just a super example of everything that's good about Canadians. Mm. He's modest. He's intellectually rigorous. He's well trained, and just generally comes across as a good, honest guy trying to do the right thing. So I just want to say that's where I come from in terms of Canada, and the people that I've known in Canada, and my wife is known in Canada. So we like you guys a lot, and thank you for being who you are. So the big things. I'm concerned. Okay, I'm going to tell a story. I like to tell stories. I was on the phone with a Canadian physician a few weeks ago for 2 hours up until midnight, sharing with me and with another person on the phone their stories of what it's like to be trying to treat COVID disease and talk about alternative treatments in Canada, and talking about what he is experiencing and perceiving regarding the vaccines and what he's seeing in his own patient populations. So a practicing physician that might have a few hundred clients, will only see the very frequent adverse events. They won't see the rare things in general. They might, but the things that happen 1 in 1,000 cases, or 1 in 10,000, or 1 in 100,000, they're not going to see those things. So they just see the average stuff. And this was a frontline GP doc just trying to take care of his patients. And he was sharing story after story of adverse events, which is the fancy word we use for sick, soreness, sickness, pain, and things like that, joint aches and more serious things that might happen after you receive a drug. And in this case, it was after receiving vaccine. And he was telling me the story. I was listening to his stories about how he would report these things within the Canadian system, things that he, as the practicing physician managing these patients, thought that they really looked like they were vaccine associated. And he would report them into the system, and they would immediately come back as being determined to not be vaccine related without any further investigation. So this really bothered him because he's a conscientious doc trying to take care of his patients, and things didn't seem right. And then he told the stories about, and I haven't confirmed them, but I hear that they're true, about some provinces having campaigns to try to get children vaccinated by enticing them with ice cream and not thinking that they had to get parental consent, and policies in Canada. And I want to be able to travel to Canada sometime, so I don't want to criticize your prime minister or anybody else, but from my training in bioethics, you really can't coerce. It's a big no-no to coerce people to take an experimental product, and that's what these vaccines are, is experimental products. So, to my way of thinking, a policy that says that the government is going to punish you by forcing you to stay on lockdown in your houses unless 70% of the population in your town accepts vaccine, that sounds like coercion. So in my training, which I've taken a lot of training in bioethics and clinical research, because I have to, because that's what I do, clinical research. Okay, so important that I behave in a bioethically acceptable fashion so that we don't have a Tuskegee or other things happen.
Yep.
And I take that job really seriously. So I think one of the things that's important to know for people is what are the fundamental bioethics surrounding experimental medicines and experimental vaccines. And there's 4 points that I'd like to touch on that can go really quick. We can elaborate on them, but to go over them quickly, point number 1, these are still experimental medical products. They're not licensed yet. We don't yet fully know how safe they are and how effective they are. Okay, so they're still experimental. Therefore, all the rules for experimentation on human subjects apply. These go back to the Nuremberg Code. Point number 2, there has to be full and complete disclosure of risks. Okay, and that means, like when you buy a medicine, it's in a little box, it's got a piece of paper inside, you open the paper, you look at it, and you see, holy moly, look at all those things that could happen to me. And there's things at the top of the list that say these happen pretty frequently. And usually they're like, I get a headache, or I have problems urinating, or something like that. And they go way down to, well, you might die, or you might, your head might explode, or whatever the thing is. Once there was a report of somebody having their head exploding, that's way down at the bottom of the list. But you have to disclose all that stuff. Okay? Same is true with experimental products. Just a minute, I have to decline a phone call. And so first there has to be full disclosure of risk. Bedrock rule. All the way down, adults can make their own decision. It's your body, it's not the state's. First rule is it has to be disclosed to you what the risks are. Second rule, that disclosure has to happen in a way that you can understand. There has to be comprehension of those risks. Okay, so I can't disclose those risks and make them all sciencey. Okay, if one of the possible risks, adverse events that may be associated with these vaccines is thrombocytopenia. Now raise your hand if you know what thrombocytopenia is.
I looked
That's
it up.
a big fancy, yeah. You looked it up. Good for you.
Yeah.
But most of your audience is going to say, what?
Yeah.
What does that mean? Okay, and it's low blood platelets. And if you have low blood platelets, you can get bleeding and clotting disorders and other things like that. And in fact, those things happen both with the disease and with the vaccines, particularly the adenoviral vector vaccines, but it looks like probably the RNA vaccines too. Okay, so we can't hide behind fancy science words. We got to stay stuff in plain language so that all of us that at least have an 8th grade level of education, I think you have a similar public school system there as we have here. So about the level that any 13-year-old that is reasonably well educated can understand. That's the level that we have to describe. Third point, fourth point in the series. So we said it's experimental products. There has to be full disclosure of risk. Those risks have to be comprehended. And here's the last point. You have to voluntarily, of your own free will, agree to take the product. Now here's in addition to all that stuff, there's one last special thing. Adolescents, children, and infants cannot give informed consent. That's why we call it the age of informed consent, the age of consent, right? And I don't know what it is in Canada, but if it's like the States, it's 18. Yep. And so up until you're 18, you can't give informed consent. That's just the rules. We've all agreed on it. When I was a 16-year-older, I would have debated you on that and said, oh, I could make my own decisions, and I darn well would try. But that's not the rules. And so there has to be informed consent of the parent or the guardian for children to accept an experimental product. This is bedrock. And part of the informed consent is you cannot coerce somebody. So if I was to write a clinical protocol for a study that was going to involve pediatric testing of a new product, and I wrote in my clinical protocol, I'm going to give out free ice cream and I don't have to get consent from the parents to do that, they would laugh me out of there. I would be, I'd be excommunicated. I'd be persona non grata. I would be Dr. Bridle subjected to that, okay? And that's just the way things are. Those are the rules. We all agree on it. And we have elaborate practices in regular clinical research to ensure that we don't end up transgressing on human rights. And then I'd like to add the last thing to your audience. You asked me to hit the key highlights.
Yep.
It's your body. We're not in an authoritarian environment in Canada or the United States, no matter what we may think. It can always get worse, trust me. And currently, we're free people for the most part. The state does not own your body. You own your body. It's your body. And my firmness on this comes from being asked. I get bombarded by questions, as you might imagine, from folks like yourselves, saying, Robert, should I do this? Should I do that? Should I accept this vaccine? Should I accept that vaccine? Should I have the second dose? Shouldn't I have the second dose? I have this medical condition. What do you think? Blah, blah, blah. And I'm not their physician. And what I resort to is it's your body. You understand your body and that background better than anybody. And, it's totally valid to communicate and get the advice of your physician or your medical provider, whoever that person is, and I encourage you to do so. But in the end, it's your body and it's your decision. And so that's the last main point I wanted to make. Now, then there's all of the jibber jabber about this toxicity or that toxicity or this adverse event and whether it's related or unrelated or whatever. And that, a lot of that's kind of scientists talking to each other or trying to talk to each other to the extent that we're allowed to in this time where there's so much censorship. And I ask your forgiveness and tolerance, but this is how we do science, is we have to talk to each other and we kind of have to intellectually arm wrestle a little bit over what is truth. And a lot of times truth isn't just one thing. As you know in your own lives, we each kind of have our own truth to a certain extent. And scientists see things differently. Some scientists come at it from different points of view. And some people in public health have the responsibility for the overall population. And so they set public policy with an eye on what's good for all Canadians. But others of us focus more on the individual. What's good for you? And there's always a tension between what's good for you and what's good for society. And my opinion is pretty strong that we can't sacrifice the rights of the individual for the broader good of society, except in rare cases. And experimental vaccines is not one of those cases.
Okay.
I'm pretty sure about that.
I want to go back because some people listening to this hear you calling them experimental vaccines over and over, and they're going, wait. I've seen the posters that say there's been no shortcuts taken. They don't get that this is an experimental vaccine. Can you explain why it, in fact, is an experimental vaccine?
Okay, so I develop vaccines for a living. And I've got all this training and stuff about how to do that, and all the steps that are required, and the regulatory steps, and all that stuff. In the normal case, when the wolf is not at the door and we have a crisis, in the normal case, so let's say a new pediatric vaccine and a new adult vaccine, usually it takes at least a decade and somewhere in the region, US dollars, of 1 to 5 billion. Okay? So it takes a decade and it takes a gob of money. And the norm at the FDA, and I'm pretty sure that the public health authorities in Canada have the same general policy, for a new vaccine, this is just the most overt example that everybody can understand. We can go into the weeds and talk about this, non-clinical toxicology or whatever, but at the big picture, the standard practice is that you have to take the vaccine to the point where at least 3,000 people have received the vaccine. They volunteered to participate in the clinical trials for the greater good, bless their hearts, okay? And so at least 3,000 people have accepted the vaccine, and they are typically followed for at least 2 years to see if anything bad happens. Because this is the honest truth. There is no perfect drug. There is no perfect vaccine. Everything comes with some risks. It's just the truth. There's no vaccine that has no risks. Ain't never happened, probably never will. And certainly these brand new technologies that haven't been widely deployed before, no one can say that there are no risks associated with it.
Right.
Nor do we really know what those risks are, because they haven't been in a lot of people for at least 2 years. What are the kinds of risks that can develop long-term? All kinds of things. The most notable is autoimmune disease. And for instance, we know with influenza, one of the most notorious side effects of influenza vaccine is very, very rare, is Guillain-Barré syndrome or paralysis of the face, of one side of the face. And I've had people that have had Guillain-Barré syndrome who hear that I'm a vaccinologist, and they come on really hard to me and quite angry about the vaccine industry, and how could I be a vaccinologist and participate in this and all this stuff, because they're justifiably angry because they've experienced this side effect. That's very rare, and the probability is that you've never encountered one of these people in your whole life. But it happens, and in the case of swine flu, it happened significantly more frequently than it happens with the regular influenza vaccine, okay? So my point is only that no vaccine is perfect. It all comes with risks. And these autoimmune syndromes that can develop if, a bunch of different ways they can happen and take a long time to develop. And other things can take a long time to develop with drugs in general. We know about thalidomide. I don't want to be scaring you with horror stories. That's not my goal. What I'm trying to say is that stuff happens. And we can't predict what it is when we're dealing with new medicines and new vaccines. And so it takes some time. So the government to say, and I've seen this and heard about this metaphor, we didn't cut corners, we just straightened out the road. And that's very glib and easy to say, but we can get into the weeds, and I guarantee you we cut corners. And one of the corners we cut is we didn't give 2 years of safety run out after administration to 3,000 people. How do I know that? You can do the calculations yourself. Has it been 2 to 3 years after we started deploying vaccine? And I'm pretty sure your math is the same as my math, and we can both conclude the answer is no. Okay, so the statement we haven't cut corners is actually false, and we can easily see how it's false. And I can go into the hardcore tech, details of this, that, and the other detail, and give you more examples. But I personally don't think it is a good idea for the government to make false statements. I think that it compromises public faith. And that's one of the most worrisome things for me as a vaccinologist, is all of this messaging that we haven't cut corners, that everything is fine, move on, don't look, there's nothing to see, when in fact, I know that there are signals out there, and most people that talk to their friends and family and the folks, and certainly the frontline docs, know that in their experience, they know of people that have had adverse events from these vaccines. And it's an odd thing because it is not scientific. It's a random walk also to say, yeah, my uncle had this thing happen. My daughter had that thing happen. And forgive me for this example. I hope I don't offend anybody. But one of the most clear-cut examples is alterations in menstrual patterns and menstrual flow in women that are not postmenopausal. And there's a lot of people that report this to their friends. Now, are they going to go online in a government database and say, oh boy, my periods were really weird afterwards and my flow was really heavy? In the scope of things that you have to do during the day, logging on to a public, to a government database and reporting about your periods is probably about 0 on your to-do list. But yet, many of these things happen, like my wife and I were really sleepy for about 4 or 5 days after we took the Moderna vaccines, and other people report this. Okay, so for the government to say, this never happens, it's all perfectly safe, well, we know from our own personal experience that isn't true. So what I say is it gets back to those bioethics.
Right.
Government kind of needs to come clean.
Right. So
on what's going on.
I was going to say, so I've been, watching online, taking in these stories from healthcare workers in the United States who have had adverse reactions, neurological, and they're connected now to multiple people. They're saying they're getting about 10 people reaching out to them daily with these reactions. Yeah. What are your thoughts on you as well?
Since that Brett Weinstein podcast, my phone doesn't, I've had to hang up 3 times while we've been talking, and my inbox is full. I get calls from crying, desperate mothers. I mean, think about that. You're out there feeding the horses, right? I have a horse farm, and you get a call. You take a phone, and you've got a distraught, crying mother who's desperate because no one will believe her. That kind of stuff just breaks your heart. As my wife is fielding a lot of that, and she notes that a lot of this traffic that I'm getting on the phone and on email and onto our website isn't people self-reporting. It's often their relatives. It's their fathers, their mothers, their husbands, their wives saying, this has happened to us. And what can I do, Dr. Malone? And it's heartbreaking. But that gets to my point, censoring all this communication, and they've all been bottled up. They can't talk about it on Facebook because they just get deleted, right? And if they talk about it to their friends say, what, are you crazy? You must be an anti-vaxxer, because we all know the government is saying this isn't happening, therefore it's not happening. And yet these people are experiencing these things. And you can imagine how frustrating and depressing that is. It's like being gaslighted by the federal government. It's kind of what it is, and it's not right. So I agree with you that we all, and I think it's kind of reaching critical mass. And it's not that I was the first to say, whoa, guys, something's wrong here. But the combination of my experience, my CV, the other things, has in this podcast that went viral, suddenly I find myself in a position where I can give voice to things and be heard that other people haven't been able to speak. And I kind of think that I have an obligation as a physician to, and as a vaccinologist, a vaccine researcher, to responsibly communicate that there's something going on here, guys, and we probably ought to pay attention to it and take care of it and stop denying it.
Let's dive into that specifically. So I'm going to put up the VAERS chart. VAERS is the United States vaccine reporting system, except there's another one as well, correct?
Yeah, VSafe is kind of kept within CDC, and they're not showing it to anybody. But little cracks are happening, like the recent MMWR report from the CDC. That's their publication, MMWR. It's an acronym, Morbidity and Mortality a Weekly Report. But you're talking about a graphic that summarizes trends overall in the VAERS database for death.
Yes.
And so let's pick that apart just a little bit, because that graph and that presentation have been criticized by many people as not being representative. And all of us know what happens on social media. Everybody's an expert and everybody nitpicks everything based on what their own frame of reference is, okay? That graph is not perfect. It is gross data. It's not normalized. It's not filtered. It hasn't been processed. It's just total gross reported events, both adverse events, and I think the one you're probably going to show is deaths.
Yes.
And the VAERS database, by definition, is a vaccine adverse event reporting system. That's the acronym. And so presumably all the reports that come into VAERS from physicians and individuals are coming in because they have some association in time, a temporal association with vaccination, as perceived by the person reporting it. So obviously, if you died, you're not going to self-report, but maybe your doc does, or your mother or daughter or whomever. So this comes in from a variety of sources, and somebody somewhere thought that these events were vaccine related. Now, they may or may not be. Now, what you're looking at with those data, if it's the graph I'm thinking of, is it's the cumulative reports of deaths, probably. There's another one that's the adverse events, over a long period of time across all vaccines. No person that is reasonably sophisticated about what VAERS represents would assert that all 6,000 of those deaths are vaccine related. We can't say that unless someone goes to do all the nitty-gritty detective work to determine whether they are or aren't vaccine related. Somebody somewhere thought that they were, okay? But nevertheless, when you look at that graph that you're showing now, that you're going to cut in, what we see is this really low baseline of deaths, and then suddenly there's a spike. And that's kind of hard to figure out how that happens unless there's something about vaccination. Now, maybe it's just because of the internet, and you know how it is now with everything going viral, and we know how there can be flash mobs in social media that's driving that. We don't know, but I think that it's fair, and I think that we can probably all agree that that graph looks like there's something there that ought to be investigated. And I think that's the responsible place to hang out, is say, doesn't look quite right. We ought to look further. And 6,000 deaths here and 30,000 deaths in Europe that have a association in time with receipt of vaccine sounds like the kind of thing we ought to look at pretty hard. Does it mean that vaccines causing death? Some people infer that. I don't. I'm a professional, is what I do for a living. I live in the world of trying to do evidence-based medicine and be rigorous. But even I agree, that looks like something that ought to be taken seriously. I don't think you need to be a genius physician scientist steeped in a stupid long amount of postgraduate training to say, that looks like a pattern. We ought to look into it. So I submit to your audience, once again, if it walks like a duck and it quacks like a duck, there's a good chance it's a duck, and we ought to make sure it's a duck, and you understand the metaphor, I'm sure.
Completely. Okay, I want to dive in a little bit. If we can just give a concise rundown of how the mRNA is intended to work, and now with new things coming forward, the study from the Japanese government, the biodistribution chart, what might be going on?
So there's 3 sources of data, and this is going to, you wanted me to be concise. Okay, so I'm going to try to be the really quick version, and then you can ask me questions.
Perfect.
There's 3 key sources of information that have some people concerned, including me. Okay, it's concerned, not alarmed, not saying that the house is on fire. I'm saying kind of looks like there's some smoke there. We better figure it out and put the fire out if it's there. Okay, so there's 3 things. One of them is this group of Canadian physicians and scientists, including Dr. Bridle, concerned about what they thought might be adverse events associated with vaccination, submitted a Freedom of Information Act to the Japanese regulatory authorities concerning what we call common technical document or initial new drug application submitted by Pfizer to the Japanese regulatory authorities to justify the administration of the Pfizer vaccine in Japan. Now, this word common technical document and initial new drug application, those are 2 word strings. What they mean is this is the package of documents. It's the report that Pfizer had to prepare that summarized everything they knew about the product, its characterization, its manufacturing, everything they knew, all the tests that they did in rats and mice and monkeys or anything else. And it is the package that the developer or the sponsor, is a technical term, the company or person that's trying to advance a new product. It's the package that person has to submit to say, hey, government regulatory authority, this is all we know, and we think this is enough to justify taking this into humans. That's what it is. So it's not a Japanese study. It's a Pfizer data package that was submitted to the Japanese government. Now, I'm going to unpack the common technical document. So what that means is that these days, we kind of have a treaty between all the major regulatory authorities worldwide that says that, hey, if you're a developer, if you use this format, we'll accept your package, and you don't have to change everything around for us and then change everything around for the European Medicines Agency and change everything around for PHAC or whatever your regulatory authority is, etc. you can just use one common document. So presumably Pfizer submitted something very similar to the Canadian regulatory authorities, the US regulatory authorities, and the European Medicines Agency. So Dr. Bridle and others were able to obtain what is usually closely held and protected and cannot be obtained through Freedom of Information Act. For some reason, the Japanese regulatory authorities have some policy, I'm ignorant of that, that allowed somebody to do a Freedom of Information Act and obtain what is otherwise typically not obtainable. It's considered privileged information and you can't FOIA it. Now I'm going to talk about, so that's one data package.
Yes.
Another group of data that I was asked to evaluate is the summary letter provided by the European Medicines Agency. Upon review of presumably that same data packet, but it was in English probably, maybe French, I don't know, probably English. So the EMA published, as is their practice, a summary letter with what their findings and determination was and justification for allowing the Pfizer vaccine to be used under EMA guidance. Okay, so that's a complementary group of information. It's a separate document in which they say Pfizer did these things and those things, and this is what we generally conclude. But it's not the same as actually getting the stuff in your hands, the stuff that Pfizer submitted. So that's another one. The third key document in this whole discussion is this now peer-reviewed and published paper from Harvard and Brigham in this nurses study that showed the levels of circulating free spike protein after receipt of Moderna vaccine, which is the highest dose of the RNA vaccines. Okay, so three things. Now we're going to unpack those a little bit. Is this good? So good. Those are the three things. It's hard for me to say this really quickly because it's complicated.
It's complicated, and I will also, because I think I have I have one of those for sure saved. I'll get those actual documents and I will put them in the post as well, because people can look at them if they like.
Awesome. Perfect statement. And by the way, I happen to have the Pfizer clinical protocol. If you want a copy of that, just email me and I'll get that to you too.
Beautiful.
Okay, it came to me I'm getting stuff from all over the world from physicians and scientists, and somebody sent me that, which was kind of amazing. Normally those things aren't disclosed. So I have the actual protocol. You can read all the documents, make your own decision. So let's unpack what the Japanese data, data package showed. That, and I received this about a day after the Canadian group received it. And I was asked by a US news website called Trial Site News to review that and the EMA document. So that's how I got involved in all this a few weeks ago. So I made a completely independent assessment of what Dr. Bridle famously put out as his assessment. And by the way, I came to exactly the same conclusions independently. And more so, I then was alarmed and concerned about what I was seeing in the Japanese data packet and the EMA summary. And so I said, I got to be sure that I'm not overreacting to this and that I've really properly processed this. And so I sent it to a colleague that I very much respect that is even more experienced in regulatory affairs. He's the president of a large European society, and I'm not going to disclose it because it would disclose, he wishes to remain anonymous, and I wish to respect that, okay? But he knows his stuff. He's been doing this for a long time. So he read the same things. He came to the same conclusions and said, oh, by the way, there's these other things that you missed. And I said, oh, thank you very much. Okay, so that's what went into the article on Trial Site News that really parallels a lot of what Dr. Bridle is saying independently, and frankly, much more eloquently. He put together, I'm in deep admiration for the precision and accuracy of the document that he put out. It's much better than what I put out. I didn't have time to do that. But he's clearly, he and his group are very concerned, and they're taking this very seriously. They're not, this is not a trivial, casual thing for them. So what does the Japanese document show and what doesn't show? One of the things about the Japanese document is that it's in Japanese. I don't read Japanese. I don't know about you. So it's in the characters. So there's a whole lot of things there that I can't read. But there's some key tables and figures that are in English. They're labeled in English, and you've seen those tables, and you may be about to project one of those tables. The one that folks don't really focus on is the figure that involves the animal studies on luciferase expression, which is really a key part of that same information bolus. So what's got folks alarmed a bit, and I'm going to say a caveat, Pfizer has submitted an updated data package to the FDA about 3 weeks ago, and presumably that must, I hope, that Pfizer, Pfizer is not a stupid company. They know what is required. And I'm presuming that the deficiencies in the Japanese data packet and in the EMA summary reflect Pfizer submitting partial data because everything was so rushed. Let's give them the benefit of the doubt.
Gotcha.
Okay.
Yep.
And they've submitted something new, and I talked to the director of Center for Biologics Evaluation and Research, Peter Marks, which is one of the big divisions of the FDA, about my concerns, and they also happen to be Dr. Bridle's concerns. And he basically said, I'm paraphrasing, we have a new data package. I'm not aware of anything in there that's particularly alarming, but please give us the time to do our job and evaluate it. And so I personally, as a regulatory professional, feel like I kind of got to give Dr. Marks and his team the respect of letting them do their job and not banging on them about stuff before they even have a chance to do it. So I want to say that about the Japanese data package is probably dated. It's a snapshot of where things were at when Pfizer was asking for permission to start human clinical trials. However, here's what the data show in the table that you're going to be showing us is on the left side of the table, there's information on, you'll see there's a stack of listing of different organs and parts of the body. And on the right also. On the right side, those are summaries of the distribution of tritiated, which is radio-tagged RNA. It has been formulated in the same way that the Pfizer vaccine is formulated. Okay, so that one on the right side shows where the RNA went in these limited numbers of rodents. In this study that is non-GLP, that's fancy regulatory speak for, it was kind of done at the level of rigor of what a graduate student or a postdoc might do at the University of Toronto or McMaster or whatever. But it wasn't done at the level of rigor of a professional toxicology lab with a board certified toxicologist, which is under quality control and all that stuff. Okay, so that's good laboratory practices. These are non-GLP studies, which is kind of the stuff that a university could do on the bench. Okay, so not very rigorous. The RNA, and then on the left panel that you're looking at, that's a summary of the lipids, the fat part. I mentioned at the start that the way that these RNA vaccines work is you take the RNA and you coat it with fat, special kinds of lipids that when they're in the right special balance can enable the RNA to get into cells and be loaded onto ribosomes and make protein like spike, okay? So the lipid is the secret sauce. And oh, by the way, this is a source of Canadian pride. I'm going to give a shout out to Dr. Peter Cullis and his team at the University of British Columbia and the company that they've created called Acuitas, which is responsible after his 40-year journey as one of the top liposome specialists in the world. They are the ones that made the technology and discovered the compounds that is making all this possible. They're like me. Peter is not somebody who seeks the limelight. But I think he and his team are unheralded heroes here. The materials and technology formulations that they created is used directly by BioNTech Pfizer and by CureVac. And the Moderna product uses basically the same stuff, but they reversed engineered a little bit to get around the patents. Okay, so yay for Canada, yay for UBC, yay for Peter Cullis. So these fats are summarized in the left-hand part of the table and where those go after they've been injected into a rodent model. And that requires special assays to be able to track those novel lipids, but they've done those assays. And those things, one of the big messages is, unlike a regular vaccine, an old school vaccine like mumps or something like that, measles, rubella, flu. Flu is perhaps the best example, okay? When you get your flu shot, for the most part, it stays put, unless you're taking the intranasal AstraZeneca product. Okay, that's a different thing. But if you get the shot injection, it kind of stays put in the place where it goes, and it gets sucked up into the lymph nodes that are in that area, and all the magic of the immune response happens. We don't have to go into that. So normally you would expect a vaccine that's injected to kind of stay put mostly. The radio-labeled RNA shows that it doesn't stay put. Much of it does, but much of it goes all over in the body. Small amount concentrates in both testis and ovaries, but also goes to spleen, liver, and other places you'd expect it to go. In contrast, the lipid part, so this is a nuance that a lot of people miss.
Yes.
The lipid part accumulates in the ovaries of this small number of rodents. Okay, so again, caveat, not a rigorous study, small number of rodents, not humans. We can go on and on, okay, lots of caveats. However, about 20% of the lipids in this study end up in the ovaries. And that is not true in the testis. Now, my friend who's a urologist that I work with, Dr. Kevin Tamara, has put on his thinking cap, and he thinks it's about Sertoli cells protecting the testis. But whatever it is, there's something different between testis and ovaries, and these lipids are accumulating in ovaries. So who cares? Well, pretty much anybody that's of reproductive age cares about this, because these are basically novel drugs. And so that's kind of a, I'm concerned moment, okay? And now, if you take that, and you'll see in the clinical protocol, Pfizer says flat out, we have not done the reproductive toxicology studies. And if you look at the EMA package, they talk about what reproductive toxicology studies were done. They don't talk about the ones that weren't done that are normally required. Genotoxicity studies have not been done. That means, can this stuff damage your DNA in any way within the assays that we normally perform? Those weren't performed. Okay, so in aggregate, a alert, educated person like yourself might look at these data and say, I'm starting to get a little worried here. Now, why would you get worried about some lipids accumulating in the ovaries of a rodent?
So can I just clarify here, the audience, really quick, Dr. Malone? There's kind of three things going on. There's the lipids, which is like the package that the mRNA is encased in. Is that correct?
Yep.
And then there's the spike proteins that your body produces after the mRNA tells the cells what to do. Correct?
And then there's the RNA itself.
And then it's the RNA.
Precisely. Okay.
Okay, so what we're talking about is actually the packaging being found in the ovaries.
Yeah, so it's equivalent. I've heard the metaphor, this is the FedEx letter package.
Yes.
Not the letter. The letter, if in the metaphor of a FedEx package with a letter in it, the letter would be the RNA, the package would be the lipids that Peter Cullis and team developed. And they're novel. They've never been in humans before at this level, a little bit in some cancer trials. And so if the rats or mice, if the rodents are predictive of humans, and remember proportionally, the human dose is smaller by weight. and our ovaries are bigger than those of a rodent. Yay. You know, you have to, once again, you got to look at these data with caution. Don't overinterpret them. But there is a history of small molecule drugs causing problems with reproductive health and birth defects. Does that mean it's going to happen? Absolutely not. Could it happen? It's happened before. So we have to be aware of that possibility, and we have to do really rigorous testing to reduce the probability. And what's that rigorous testing that we have to do? Well, GLP reproductive toxicology studies, which, by the way, weren't done. So now we're even a little more worried. Now it gets a little bit even more squirrely, if you use that expression in Canada, complicated. The RNA that they used for these tests was not the spike RNA.
Okay.
It was the RNA that codes for the thing that makes the firefly tail glow. I think you've got fireflies in Canada. And so the thing that makes the firefly tail glow is a protein called luciferase. And those of us that do this for a living use that protein as a way to detect whether or not a RNA or DNA or a gene transfer is successful. Because if we use that as what we call a reporter gene, then that protein, if we give the cells or tissues, or we can inject it in the mouse, if we give them the substrate that the firefly uses, it's called luciferin. That protein can cut that luciferin and do a chemical reaction that releases a photon of light, a little teeny tiny flash of light. And we have really cool technology for detecting light called photomultiplier tubes, super sensitive. Now, those photomultiplier tubes can be used to assay and extract, grind up the tissue and make a solution of it and add a little tiny bit to a test tube and put it in the machine and see whether or not how much light we get made over what period of time. And that can give us a really linear estimate of exactly how much luciferase protein is made. It turns out that the assay is that the method is so good that you can take a whole animal, anesthetized or, sacrifice it, and put it on a laboratory bench in a darkroom and inject luciferin into its abdomen intraperitoneally. And the animal will start glowing in those places that have luciferase protein. Now, this is a pretty insensitive way. This is really low sensitivity because those photons have got to get out of that tissue, go all the way through the muscle and fat and everything else, through the skin, through the hair, and hit the photomultiplier tube if it's an intact animal. So it's the least sensitive way to detect whether or not the distribution of gene expression that might happen. But this happens to be the method that Pfizer used to say that the gene expression from the surrogate RNA, not the real one, is localized only to the site of expression. Ooh, that sounds kind of like somebody's cooking the books a little bit, right? You follow what I'm saying? Because
Right.
they could have dissected those animals and pulled out the ovaries and the testis and the kidneys and the liver and put it in the test tube, ground it up, got a precise number, but they didn't. They looked at the total animal, which is far less sensitive. And what it'll do is it'll give you a signal where it'll tell you where most of the expression is, but not everything. So what you end up here with this data package is a series of things that weren't done quite right. And this gets back to the poster and the graphic that you're talking about. No shortcuts, we only straightened out the road. Well, I'm sorry, but that's not true. So that's that data package. It's got three things it's looking at: the distribution of expression, but it's not actually using the drug. It's not looking at spike expression. It's looking at luciferase, a surrogate, radio-labeled RNA distribution. That's more solid, but it's only in a small number of animals. It's non-GLP. And the distribution of the lipids. And the distribution of lipids has some things on there that look a little bit concerning, particularly about reproductive tissue, which is something that we're particularly sensitive to because it's not just today, it's our children. Okay, so that's that data package. And it's supported by the summary letter from EMA. Points out many of the same things, but without all the nuance. Then we have the women and nurses study, and I'm going to segue to that. There's a truism among those of us that have done animal research, of which I'm one. I used to be a primate center investigator. I'm now a vegetarian. I don't sacrifice animals anymore, but once upon a time early in my career, I sacrificed many, many, many mice, so I'm familiar with this. One of the things about animal research is that we all of those that do it recognize, we have a saying, mice lie, monkeys mislead. The only thing that predicts response in humans is response in humans. Everything else is a rough estimate that we use to try to justify starting our human studies. But as soon as we start getting actual human data, all that stuff we kind of throw in the trash can a little bit, because what matters is what actually happens in humans.
Yes.
And we can't always predict what that's going to be. So the nurses study is the example of actually doing it in humans. And the people that engineered the Moderna vaccine, which are the people at the Vaccine Research Center at NIH, a lot of people say, oh, that's the Moderna product. No, actually, it's the NIH product that Moderna sells, but it was designed and engineered at NIH. And the formulations were optimized at a company called Ginkgo. They weren't actually done by Moderna. So, you know, Moderna is kind of has the money and the organization, and they're funded by DARPA and all that good stuff. And we could go down that rabbit hole, but it's really the NIH product. And they engineered the spike, which is the antigen. I think we talked a little bit about spike earlier. So spike is this protein complex that you'll see in pictures of the virus that kind of sticks out as a knob. And that makes it a good antigen for getting an immune response. And it would be really good if we had super duper antibodies that block the receptor binding domain of spike so that the viruses can't infect cells. That would be good. And we have some of those. Turns out they don't stop the disease very well. But nevertheless, that was the logic, is they engineered spike protein as the antigen into the RNA. And they added some extra stuff. They engineered spike so that it stays in an open, it's locked in an open conformation so that the receptor binding domain can easily be accessed. Okay? Now, some people say they did that for safety, but that's a bit of a paradox because back then they didn't know that there was a safety problem with spike. So it's a little circular to say they did it for safety when they didn't really know that safety was going to be a problem back then. And another thing they did was they engineered it so it had a little tail on it, membrane, transmembrane domain, that makes it so that when it gets made in your cells, it gets exported to the surface of the cell, and it kind of stays there. It gets stuck in the membrane and is supposed to stay there. What the nurses, and they did some studies and they said, hey, this all works, it's great, let's go on and do it in humans. It turns out from the nurses study that in humans, it doesn't work that way. And the spike gets cleaved off by a protease, unknown, and it ends up in the circulation. Now, is the spike that's in circulation in the blood the same structure, the same folding, etc., as the spike that we know from many peer-reviewed publications is toxic and does open the blood-brain barrier? That's all settled literature. It's very clear. But can we say that the spike that's released after immunization and was found floating in the blood of these nurses for 14 to 28 days, basically 2 weeks to a month, at non-trivial levels, can we say for sure that that spike is more safe or less safe than the spike that is naturally produced by the virus? Those studies may have been done. I hope that they were done. I hope that they're in the Pfizer data package that the FDA is looking at right now, but I haven't seen them and I haven't seen them published, okay? So the way things work is that if the pharmaceutical company or the NIH is going to re-engineer a protein that they're going to express from a gene therapy vector in your body or my body, it's their job to prove it's safe. we don't start with the assumption, oh, it must be safe because NIH did it.
Right.
You know, you have to prove it. That's the way this works. Is the assumption is that it's not safe until proven safe. Just the rules, and it makes sense. Most of us can understand why that's the case, because the job number one of the regulatory authorities, above all else, is to make sure that we have unadulterated, effective, safe products. Okay, and so safe, underscored, and it's particularly true for vaccines, because unlike cancer, when you may be dying a year from now, and so if you take a drug that might cause you to be more likely to die 5 years from now, but you survive past this first year, that's probably okay. You might say that's an okay thing for me. But a vaccine is being administered to somebody that's healthy, that may or may not eventually get exposed to COVID, and to SARS-CoV-2. They may or may not at some point in the future, unknown, develop COVID, but at the point when they take the vaccine, they're healthy. Okay, and so that's the rule with vaccines. They have to be ultra safe. Okay, so it's the burden is absolutely on the developer to prove safety. So there's a whole argument on the internet about whether or not the engineered spike is safe, and and that stating that, well, all that other stuff with the regular spike, that doesn't matter because yes, okay, we agree, it opens the blood-brain barrier, it's toxic, the data's there, the Salk Institute, blah blah blah. But some people assert, oh, the great minds at NIH, and there are many great minds there, engineered this so that it's perfectly safe. The problem with that is that at the time they were doing the engineering, there was nobody saying that spike isn't safe. Those data came out later.
Right.
Okay? But they have the job of proving that it's safe by doing the, wait for it, GLP toxicology studies. Okay, that's what we do. And they weren't done. Okay, so does that mean that we have a problem? From where I stand, we have a risk. We don't know how bad that risk is. We don't know whether it's manifest, but we have a risk, and we have to address it seriously.
Okay, can I just clarify? Because so there's two things that you went through. There's the organ biodistribution. So there's a little bit of a uh-oh when we have these this packaging accumulating in the ovaries and in the bones. That's in the chart as well. That's kind of like a signal.
Yes.
And then, oh, it says my internet's unstable. Let's just wait a second. And then you have.
Yes.
Can you hear me now? Am I good?
Yes, you're coming in and out, but let's carry on. We'll fake it till we make it.
And I'm gonna splice this up a bit and put other stuff over it too. And then we have another concern, which is the spike actually getting in the bloodstream. So that's been proven with this.
Right.
That's been proven that is happening. But the question is, people are arguing whether or not that's an issue or not. Some are saying it's perfectly safe. Some are saying it's not.
Just so. And that's an appropriate academic argument to have, and we have a way to solve that argument, resolve it. And we can argue about it until the cows come home, but at some point, somebody's going to have to do some science and test it. And in the meantime, what we have are these adverse event signals. And things like brain fog or neuropathies or things that could be explained by disruption of blood-brain barrier or neural membrane integrity. And we have for sure the blood clotting problem. And we have blood clots in the brain. And for a while they put J&J vaccine on hold and the Norwegians won't even touch it. And I can tell you the people in Taiwan are pretty aggravated because they're having deaths in their elders, which socially in Taiwan is a big problem because they have huge respect for their elders. And so we know that the adenoviral vectors expressing spike are causing blood coagulation problems. We know that spike causes blood coagulation problems. Here's the thing about all of this. The active drug substance is the spike. The spike is what's making the immune response happen. When you take a traditional vaccine, the regulatory authorities make you characterize precisely how much of that protein is going into the body. But with the gene therapy approaches, the agencies did not require that the developers determine how much of the spike protein is being produced by your own cellular factories. So the truth is, the kind of inconvenient truth here, is that no one really knows how much spike protein is being produced.
I was going to say,
with the gene therapy strategy.
How do you have an off switch?
With the RNA, the off switch is that the RNA degrades.
Okay.
But with the adenoviral vectors, they persist for a much longer period. So that may explain why the signal to noise ratio for clotting in the adenoviral vectors and some of the other problems is much stronger than it is with the RNA. That doesn't mean that the RNA is causing the same problems, but it also doesn't rule out that it's not causing those problems.
Okay. Now there's a
and
lot.
Here's the kind of where the rub really gets at you.
Yep.
Is, as you've noticed, some of the symptoms that people are reporting are very similar to some of the symptoms of COVID.
Yes.
And some of the symptoms of long COVID. And some doctors seem to be finding that the lab tests, blood lab tests about how your C-reactive protein changes and things like that, in people that have this kind of post-genetic vaccination syndrome, seem remarkably similar to the lab tests of people that have long COVID. And so that's another thing that it doesn't mean for sure that spike is causing this in both situations, but this is another one of those that walks like a duck and quacks like a duck and kind of looks like a duck. So it would make sense out of things that otherwise don't make sense. So it's a viable hypothesis, or sciencey talk, that spike is associated with these common adverse events, both in the disease, more severe in the disease because a lot more spike, but also with these vaccines. And that's another data point, another safety signal, another hypothesis, another concern. And here's the thing, all of this stuff really should have been figured out before it went to humans. It's a little bit alarming to me as a professional to find out, many months in from this Harvard nurses study, that this protein is being cut loose from the surface of cells and circulating around in the blood. That's the kind of thing we should have been able to know before it ever went into a single human arm. So that, again, did they just straighten out the road or did they cut some corners? I don't want to say, but it kind of looks like there was some corners cut and that they mattered. So that doesn't inspire confidence. And it gets back to my point, does the vaccine save lives? Do these vaccines save lives? My opinion, yes. Okay. I personally feel like no matter what happens with the Nobel Prize or anything else, whether people completely forget about me and write me out of history, or they say, "Oh, Robert, you were the guy." Okay, whatever happens there, I can go to my grave and know in my heart that I contributed to saving the lives of a bunch of people by helping initiate this technology. So I believe firmly that lives have been saved, but that doesn't get out of jail free card to say that we ignore the safety risks. And it gets back to the bioethics. Rule number 1, full and complete disclosure of risks so that you can make your own informed decision because you are an intelligent adult. And the rules are that you have to have the information so you can, now, if you decide not to use that information, that's your choice. But from where I sit, and I'm going to say something kind of strong. Hopefully I can still travel to Canada. From where I sit, it's the job of the government to persuade the population, not to compel the population. It's their job to show us the information that an informed person like yourself can look at and say, okay, I understand the risk benefit, and I'm going to take this vaccine because I think it's the best for me. Or I'm going to take that vaccine, or I'm going to decide to take no vaccine and walk around with a mask for the rest of my life. Whatever your decision is, it's been grounded in full and complete disclosure of risks and comprehension of those risks, because those are the rules. And that's my position. I'm not saying that this is toxic vaccines, they're going to kill you, or any of those things that have been attributed to me. I'm just saying you have the right to understand and make your own decision.
And in your opinion, we haven't been given the information to fully understand?
That's my opinion. And part of it is just because of the system. There hasn't been enough time. And we're still learning about what these adverse events are. They're still coming out. So the ones that I hear about from my friends at the FDA that aren't yet publicly disclosed, now we know the cardiomyopathy signal. That's been finally disclosed, yay. There's another safety signal about thrombocytopenia that may or may not be real. It's not really disclosed yet. Another one that seems to be embedded in there is reactivation of latent viruses. So these are the people that have shingles crop up right after they get vaccinated. Okay, now shingles is not something you can ignore. Trust me, I've had it. It hurts. Okay, and it happens at a certain random frequency when our bodies get stressed or whatever. But there does seem to be an association with virus reactivation, and it may not just be shingles. There are other viruses that hide in our genome, in our, nerve systems and ganglia and all that kind of stuff. And something about the RNA vaccine preparations seem to be kind of lighting up a danger signal and causing some of these viruses to jump out and do bad stuff. And we don't understand it. And is it real or is it not real? So those are three examples. The dysmenorrhea seems to be really common. The brain fog seems to be really common. Some of these neuropathies seem to be fairly common.
What's dysmenorrhea? What's dysmenorrhea?
I'm sorry, that's the fancy word for your menses aren't regular.
Oh.
Or normal.
Okay, so I should.
Sorry about that.
No, it's okay.
Yeah, sorry. I got doctor-y, science-y on you. Forgive me. So there are signals out there. I know they exist. And I don't know how frequently they happen. I don't know how severe they are in the range of severity. See, the thing with some of these, because we don't know, think it through with me, okay? Just walk with me for a moment.
Yeah.
These are gene therapy technologies. They cause your cells to start making a foreign protein. That foreign protein isn't just an antigen, it also has biologic effects. Okay, how much of those biologic effects, I don't know. No, is the engineered ones different from the wild type? To be determined. But nobody characterized how much of those proteins are being expressed. And what that means is that when you get a jab, the amount of protein that you make, spike protein, may be very different from what your neighbor makes.
Right.
And so it could be that if we have a bell curve and you're on the side of the curve of my body didn't make a whole lot of protein, that you get an immune response, but you don't get a lot of this other stuff. And it could be that your neighbor that's on the other side of the bell curve had a really good jab and they made a ton of protein and they had more severe adverse events. If there's a linear relationship between dosage and adverse event, but we can't know that because no one characterized it. You know, is it plausible? I think so. Is it plausible enough that I think that it merits more rigorous analysis? I think so. Is it plausible enough that your neighbors and friends and mother and father and your nieces and nephews ought to be informed that this could be going on? I feel really strongly that's the case. So that's where I'm at. I don't want to sound like an alarmist. I'm not an anti-vaxxer. I do believe these things are saving lives, but I also believe that the pharmaceutical companies and the regulatory agencies have a statutory obligation for full and complete disclosure, and a kind of and truly a moral obligation. And so that's when you know, if somebody was to walk up to the street and say, Robert, why are you raising all this kerfuffle? I would say, because it's the right thing to do, because I believe strongly as a physician and as a scientist that I have kind of an moral obligation, and I know that this is where Dr. Bridle is coming from. As experts, we are able to see patterns and associations that aren't necessarily obvious to everybody. And we believe that we have a moral obligation, truly. I mean, do you think that Dr. Bridle is doing this because he thinks he's going to get a pay raise from it? You know, he knew that he's putting his career at risk, that his family may have to eat pork and beans in a couple of months because he's going to lose his grant funding. Now, if you're facing that, are you going to say, hey, I'm going to write this big thing and get everybody pissed off at me and try to take away my money? No, you're not going to do that. The only reason you're going to do it is if you really believe strongly that there's something going on here, and you have a moral obligation to alert people to the possibility that something isn't right. And I think he deserves a prize. I think he deserves a great big hug from the Canadian people for being a good guy. That's my opinion. Let alone being attacked on the internet. It's just wrong. And it's how science has to progress. We have to be allowed to talk to each other and say, hey, are you seeing this? Does this worry you? What do you think about this? How do you interpret this paper, this document that we just got out of Japan? What do you think about it? We have to be allowed to do that if science is going to work.
So there's a lot of people...
thanks for listening to that.
No, thank you. And we'll just chat a little bit about this because I think it's so important. There are people who believe that there's no censorship going on. What would you say to that?
Well, I have directly experienced it. The book that my wife primarily wrote at the first week in February on preparing and protecting yourself from the novel coronavirus, in which she advocated for mask use, among other things. Put it out as a paperback on Amazon, self-published, you know, at a very low price to cover basically Amazon's publishing expenses. Got taken down about a month and a half after it was up there for people. And Amazon would never give us an answer as to why it was pulled. So that was censorship, you know, personal. I'm just talking about my own personal walk through time. Okay. Jill's book got pulled, 100 pages. She busted her can. Imagine, we heard about this virus in the first week in January, and she got a book of 100 pages out to help people to protect themselves in the first week in February. And then it was taken down and arbitrarily, and no one would tell her why, which is against Amazon policy. So there's point number one. Mm. Then there's all the experience that myself, my colleagues, fellow academics are experiencing around trying to publish anything having to do with repurposed drugs. It's just gotten wicked hard. And you could find my own experiences as an editor of a special volume on drug repurposing in Frontiers in Pharmacology, covered in The Scientist and in Trial Site News. So there's another data point. I think we're at a point where, unless you're a millennial, pretty much all of us old folks have been on Facebook. You know, we use Facebook to publicize our horse breeding operation, and my wife has I don't know, up to 9,000 people on her Facebook links. She's a big Lusitano Facebook horse fan person, and she controls these sites. And we can't post anything on Facebook that has anything to do with this virus. Our own academic publications, we can't post them. They just get taken down by Facebook right away. And then you have these things like, you know, 200,000 people that claim that they've suffered adverse events from vaccines that have self-assembled a Facebook group and all joined it to talk about what they've been experiencing. And Facebook decides to delete the group and all the people. Then there is the chronic examples of people being kicked off of YouTube, Facebook, LinkedIn, other things, if they talk about forbidden subjects having to do with vaccines, vaccine safety, repurposed drugs, or other things. Then, so I'm just putting logs on the fire here. Now that I've kind of come out, in a sense, I mean, I'm kind of out of the closet because of the Brett Weinstein podcast going viral. I'm having scientists and physicians from all over the world send me stuff. Not only are the patients calling me, you know, with their own stories of suffering, but fellow docs are saying, "Hey, Robert, are you aware of this? Are you aware of that?" And the other thing. And I've also had direct reports from colleagues, physicians, scientists, colleagues in France that have been subjected to even more egregious censorship than the Canadian physicians have. I mean, you could go to jail in France if you're a doc and you don't toe the line. So lots of examples. But so like many of us, we've been kind of saying, how did this happen? How is this all coordinated? It just doesn't make sense. Unless you're somebody who just goes straight to conspiracy theories and thinks that somebody is the puppeteer controlling everything because they want to take over the world and make huge amounts of money or whatever. You know, there's that whole world that's kind of a, a Reddit subgroup kind of mentality. For the rest of us that try to stay grounded in reality, we're like scratching our heads. How does this happen? So a doc sent me a series of reports and documents, including a BBC press announcement, that there is a, I can send you the links. There was a arrangement in a organization set up among the major media companies in the United States, Agency France, AFP, AP, Canadian Broadcasting Corporation, CBC, Facebook, Microsoft, Twitter. It's a long list of organizations that all came together in 2019, I believe, as a group that would cooperate to control spread of political disinformation. And they all agreed that they would work together in the event of any major crisis or large election that was being perturbed by some outside force that was trying to influence it inappropriately. And the press announcement is out there from BBC, you can look it up, that this group had a meeting and they said, well, we have to do the same thing for COVID disinformation about vaccines. And so you see all of these organizations that are suppressing information and shutting down discussion on social media and other forums and in the press have all come to an agreement. They're all proud of it. They've announced it. They're doing it for the common good, they believe. And they are stifling communication, including between scientists, by the way. And it's all for the best reasons. The best laid plans of mice and men often go awry. And they think they're doing good work by suppressing disinformation about vaccines, but the scientific community is a little bit alarmed. And so, for instance, Brett, just to close with another example, Brett Weinstein, that runs this Dark Horse podcast, which is what caused you to contact me in the first place, prior to interviewing me, he had interviewed Pierre Corey, who is the, I think, kind of over-the-top advocate for ivermectin. I know Pierre. I love him. He's a great person. He's incredibly enthusiastic, and his style is not my style, in case you didn't figure that out. He's unabashedly a strong-willed, passionate New Yorker, and he doesn't apologize for it, and he can swear up a storm just as much as any farmhand. We'll leave it at that. So Brett had Pierre, not that Pierre was swearing, but had him on his podcast talking about ivermectin, and Brett is not a big fan. And after 100,000 views on his podcast on YouTube, that got deleted, and he got a black mark from YouTube. Then the following week, we did the podcast with me and Brett and Steve, and as you've seen, Steve is a little bit cut from the Pierre Corey cloth. He's very enthusiastic about fluvoxamine, ivermectin, and very passionate about what he as an engineer has encountered in terms of adverse events with vaccines. I stayed really tightly controlled with the message of staying on track and on message about safety, bioethics, and evidence-based medicine. And I'm told by many people that it's a fascinating conversation that goes on for 3 hours. Who would listen to a podcast for 3 hours? Well, almost 800,000 people over the span of a week. Crazy numbers. It all went viral. My life will probably never be the same. After about a week and about 800,000 people, YouTube took it down, and he got black mark number 2. And he cannot, now he's at risk of losing his income from running his podcasts, because these things can make money. Secret here, from advertising. If he does another wrong step. Now, there's some good news. I don't know if he's announced this today when he was on another podcast down in Austin. It hasn't been revealed yet. It hasn't gone up. He told me this morning, I rolled out of bed at 7:30, and he asked me to coach him on a bunch of data and information that he was going to talk about in today's podcast. It's being run by another person that I won't advertise for right now. But in any case, he told me that now YouTube has come to him and wants to have a conversation about censorship because of the Matt Taibbi article that came out that made Brett Weinstein the face of the censorship that's going on. So does censorship happen? Yes. Does it happen to academics? Yes. Does it happen to average folk? All you got to do is go on to Facebook and try to post something about vaccines or ivermectin and see what happens to you. So you can verify, you can do your own little experiment to test whether or not this is true. You don't have to believe anybody else. I encourage you to verify yourself. Don't take anybody else's word for it. Try it yourself, and I think you'll find out what we've all been encountering. And in fact, it's not a crazy conspiracy theory. It's announced in a press release from the British Broadcasting Corporation that they're doing this, okay? And this is the group that's doing it, and they're all very above board, and they think they're doing it for the right reasons. So yes, it happens. It's happened. It's gotten kind of problem bad, and it's stifling scientific discussions. It's hurting people that just have a need to tell their story to each other. And I think it's wrong, and it's not well thought out. And here's the rub, really. If they're going to do this now, what next are they going to do it for? So I'll leave you with that on the censorship.
Yeah, I mean, I even told you that people were quite upset when I started questioning what's going on. And I've even seen, unfriending me, unfollowing me, and I just want to have the conversation. Let's just have the conversation.
Yeah.
Okay,
I'm
some
with
people
you.
sent me some questions. One was, what are your thoughts in Canada? People who got the AstraZeneca aren't able to get a second dose of AstraZeneca, so they're mixing and matching vaccines here. What are your, if you can speak to that, what are your thoughts on that?
Okay, I'll be glad to. One of the things that may be discussed at the upcoming ACIP meeting is whether or not a single dose is going to be adequate. The AZ vaccine is, in the slang we use, it's relatively hot. You want a hot vaccine? Yellow fever vaccine is hot. And the Ebola vaccine that the PHAC created that we now call the Merck Ebola vaccine, yay, another win for Canada. Well done. That is a hot vaccine. When you take that, you hurt a little bit. You know, your joints and other things feel it. Okay. So the adenoviral vectored vaccines seem to produce spike for a good long time. They are associated with some risks that aren't currently thought to exist for the RNA vaccines. And there is active discussion about whether or not those can be used as a single dose, and whether or not even the RNA vaccines might be sufficient as a single dose, and whether the RNA vaccines dosing needs to be dropped. And there is discussions about doing, the problem with mixing and matching is technically, from a regulatory standpoint, the only way that the regulators can say this is okay is if they have structured data that shows that it's okay. However, as a vaccinologist, we all know that one of the best ways to get a strong immune response is to mix two different vaccines. Okay, we call it a heterologous prime boost. Okay, and it's one of the ways to make a not so perfect vaccine work pretty good is to take two that are presenting the antigen slightly differently and use them in a combination in a prime boost. But to do that responsibly requires that there be appropriate clinical studies to show that, yes, it works and it doesn't cause more harm. So technically, the regulatory authorities are in a box. They can't say, yeah, it's okay. But functionally, they can't really say, don't do it, right. Usually. And I think this gets back to my point. It's your body. It's your choice. And I don't want to advise one way or the other, but there is an active discussion with all of these to just go to a single dose. And a lot of the adverse events are higher with the second dose. And I think that those that are in this position have to read, think for themselves, and make their own decisions. That's a safe place for me as a physician, not to advise you what to do. I don't want to second guess your authorities. But I do feel strongly that you have the right, you and your listeners, to make your own decisions about what you want to do with your body. But there is merit to the idea of not needing a second dose with the AstraZeneca vaccine.
Okay. Is there, in summary, what should, the people listening be considering the most right now? There's lots of ideas of, anybody who's questioning the vaccine is anti-vax, or they're stopping us from being able to live our normal lives. What is your conclusion on the information that's come across your desk?
So let me address herd immunity, which is what the government is really in their, and I'm sure in good faith. The government officials are looking at this. They want the country to be back running economically full speed ahead. So do we all, right? I don't think I doubt there are many Canadians that are saying, we really want to limp along economically for the next 3 years. I doubt that's happening. I doubt that anybody is saying, I'm not going to take vaccine because that's going to make for sure that the government is going to change because it's going to crash the economy. I doubt people are thinking that either, okay? Just kind of doesn't make sense unless you're a desperate politico, trying to craft some crazy strategy or writing a piece in a political journal. But for the most of us average people, we don't think like that. Okay. In my opinion, herd immunity per se from vaccination is unlikely to be achieved. Okay, that's an ugly truth. That's my opinion. There's a lot of people that think, oh, if we could only get herd immunity worldwide, we could eradicate this virus. I'm sorry, I've been doing this for 30 years. If that's the case, we'd have already gotten rid of polio. There's just too many places that viruses can hide. And the other thing about the whole herd immunity argument is there's a bunch of things that are kind of don't really withstand close scrutiny. One is that these rates of efficacy or effectiveness, these are the fancy scientific terms for how good the vaccine is working, are pretty much all reported as protection from severe disease or protection from hospitalization. They're all disease endpoints. The disease is a separate thing from the virus infection. The virus infection is necessary to trigger the disease, but the disease happens because of your body kind of overreacting to the infection, and some people get it worse and some people don't get it at all. So maybe 20%, maybe 50% of people that are infected don't get the disease. So if we're measuring vaccine efficacy about protection from disease, what we're not measuring is the ability of the vaccine to prevent spread of the virus. That's a whole different thing. We call that transmissibility. I'm not aware of any of these endpoints for licensure being wrapped around transmissibility. Herd immunity is about blocking transmissibility. It's not about stopping the disease incidence. Okay? So we really don't even have the data to know how many people would have to be vaccinated to stop the spread of the virus, because we're not measuring that. We're measuring stopping the disease, which is a whole separate thing. Okay? Furthermore, we know from a variety of studies that are just getting published now and have been published that the vaccines that are out there aren't stopping transmission. They may be reducing viral load and measures like that. But where the rubber hits the road is if I take the vaccine and I get exposed to virus, am I not going to get infected? Now, what we all care about is the vaccine going to keep me from going to the hospital and dying? That's an important endpoint. But in terms of herd immunity, the one that matters is the vaccine going to stop me from getting infected? Or if I do get infected just a little bit, is it going to stop me from infecting somebody else? And the data are pretty clear that the viral load and the shedding may go down a bit, but it's not stopping it. These are not sterilizing vaccines. I'm not talking about in the reproductive sense. I'm talking about sterilizing in the sense of there's no more virus replication, okay? They're not that good. Very few vaccines are, okay? They're preventing from disease, which means they're not blocking transmission, which means actually they're not that great for reaching herd immunity. Now here's the real dark and ugly thing behind that. Okay, so I'm going to say something that's going to be a little scary. But all veterinarians know this. Particularly in the Netherlands, this is kind of a staple of veterinary vaccinology. The veterinarians in much of Belgium and the Netherlands, Holland, They don't vaccinate herd animals. They don't vaccinate their chickens. They don't vaccinate their pigs. If they get a virus loose in their flocks or herds, you know, we all know this. Any of you that are farmers understand about foot and mouth disease. Okay, if you get foot and mouth disease in the herd, you cull the herd, and that's what the Dutch do with their chickens and their pigs. Now, for obvious reasons, we don't do that with humans. But the reason they do that is if you vaccinate and a virus gets loose in that flock or that herd, the virus will evolve to escape the vaccine. The only way that you can stop that is if you have such a super duper vaccine that the virus can never get started if you're vaccinated. Our vaccines aren't that good. So then what happens? Vaccines don't completely stop the virus. The virus can still replicate. Now we've got to talk a little tiny bit about virus evolution.
Yes.
Viruses are amazing little evolutionary engines. They produce huge numbers of offspring. It's like you had 3 billion children, and even 2 billion of them were defective in some way. 1 billion of your children might still survive. And if there was some selective pressure of that billion that survives, I'm talking about the numbers that would come as offspring from a virus infection, and they're actually the numbers I'm giving you are low. So you get this very large number of infectious particles that come off. A lot of them have mutations, especially in an RNA virus, and they're subjected to a new selective pressure, the immune system. And if 1 or 2 of them, or 10, manage to get around and escape, then they're likely to be selected for, replicate, spread. And what do we get from that? Well, things like the Delta variant. So there is a group of veterinarians. I'm not saying that Dr. Bridle is one of them, but particularly in Europe and the Netherlands, pardon the barking dogs, that make the point that we're setting ourselves up for evolution of escape mutants with our vaccination strategies. So when people say to me as a virologist with all of my, stupid long amount of training and years of experience, all that kind of stuff, and they say, well, we can eradicate this virus, I kind of say, good luck with that. The probability is that it's going to be more like flu, and we're going to be living with it the rest of our lives in various forms. And we kind of got to get used to the fact, which is part of why I advocate for drug development, therapeutic drug development, not just vaccines. I'm not saying we shouldn't have vaccines. I think that we have to have a coordinated approach of everything we can come up with to save people's lives and keep them. I've had COVID, okay? My body will never be the same. I will never have as much stamina. I will never have as much oxygenation. For the rest of my life, I'll probably die early. It's just the way things are. I've had long COVID. I don't recommend it. But, life is like this. We don't always get to select what happens to us. I think that we all kind of got to come to terms that the Lone Ranger isn't going to ride. The Mounties are not going to come rescue us, okay? We kind of got to deal with it. And there's some unpleasant truths. And one is that the vaccine is not perfectly protective. And I don't think there are many vaccines that are. And we're going to have these escape mutants, and the escape mutants are going to be doing their darnest to figure out how to get around the vaccines. And this is going to be our lives, just like it is with flu. So
what are some solutions?
Pardon?
What if you were to make a plan, what would you give people for solutions? What would you say we can do?
So that's an interesting question. Thank you for saying it. So I had one of these various people that contacted me was somebody representing a group of industrialists in the island of Taiwan. And I may or may not soon find myself on a jet plane going to Taiwan. Good thing I got vaccinated so that I can, travel internationally. And I've been thinking about what would I say to them, an island nation that can control its borders, kind of like New Zealand can. What would and their problem is that they are operating off of J&J, I think it is, vaccine that was donated to them by Japan. And they are losing, as I mentioned, elders from blood clotting problems. So they're kind of upset. And so I've been thinking about, what am I going to advise them? In their case, I'm probably going to advise them that they start a campaign with some of these repurposed, inexpensive drugs and that show benefit. And I don't want to name the drug here. I'm not pushing drugs. But there are drugs that are, the data are trickling in from all over the world, and they're showing that some of these drugs look like they're useful for prevention of the disease. Whether or not they're preventing viral spread, I don't have the data for that. I just wrote to somebody in the UK that is summarizing all those studies to see if he's got it. He hasn't written back. But I'm going to probably advise them to use a combination of repurposed agents in the present that are inexpensive, that can be taken chronically and safely at certain doses. And then I'm going to try to help them get access to some of the more potent, more traditional vaccines through organizations like CEPI. And roll out an integrated strategy together with protecting their borders and going into more of a island-wide quarantine environment, and enhancing personal protection. But Taiwan is a very compliant culture, so I'm sure they're all wearing masks and very dutifully keeping social distancing, because that's kind of how they're wired culturally. And I think that'll be a great opportunity to see if a strategy like that could work. Now, would a strategy like that work in a country as big as Canada? I think not. But I do think that there's merit to the idea that we use every tool at our discretion, at our availability, and don't just rely on one strategy. If there's a lesson in virology that was taught to us by AIDS, and this has been one of our guiding principles in the group that I work with at the DoD in the United States, is let's remember what happened with AIDS. Remember, I go all the way back there. I'm so old, see? And one of the lessons that came out of the whole AIDS crisis is that you need to have multiple strategies, multiple agents that are overlapping, and then you can knock the virus down, because each strategy may be able to prevent escape of 1 in 10 to the 6th viruses. But if you have 10 to the 9th viruses, that's a problem. But if you have 3 different strategies that all protect at the level of 1 in 10 to the 6th, the brilliant mathematicians in your audience will immediately say, well, that's 10 to the 6th times 10 to the 6th times 10 to the 6th. That's a big number. And that's bigger than the number of viruses. And that's how we do it. And that's how we did it with AIDS, is we had overlapping strategies that synergize in that kind of a way. And then we're able to knock it down so that people that otherwise would have been in the ground years ago are still walking and talking and participating in our culture and loving and, living their full lives because of this logic. And that's how we beat a virus, is we have multiple overlapping strategies. And I think it does us all a disservice to just put all our eggs in one basket and say that vaccines are going to cure everything, because they don't. I'm a vaccinologist. I wish that was true. I wish that the technology that I helped create 30 years ago was so fantastic that you would take one shot and it would be virus be gone, and it would never happen again, and we'd all live happily ever after. But it ain't that good. And so I think we got to be smart about it and have our eyes wide open and learn from the past and learn from AIDS and come up with multiple overlapping strategies. And I think that's the way out of the woods for most of us. But there's always going to be pockets of people in far off places that don't take vaccine that the virus is going to stick around in. And I don't think that we're going to be living with this version of a coronavirus. Certainly until I'm in the ground. Have I answered your question?
Yes. Now, is there anything else you feel like we need to cover? I feel like we went through a lot of stuff, and what I'm going to do is
We did.
I'm going to try to get this chat a little bit shorter and put some pictures and graphs and stuff over top too, so it's visually appealing. Besides our faces being visually appealing, do you feel anything has been missed?
Yeah, there's a bunch of stuff we can go on for hours.
Yes.
I was talking to Brett this morning, and he was like, "Oh, good heavens, we have to do another podcast. This is so fascinating." You know, as he's learning more about virology, it goes deep and wide. But it doesn't have to come from me. You have excellent virologists there. Dr. Bridle is one of them. There are many others. We just had an internet problem. But I think this is a pretty good start.
I think so too. And the last thing I really want to touch on, because this is what's hurt my heart, and you had an amazing chat about this, and you've alluded to it, the victims of the adverse reactions. I feel really bad for them right now.
Me too. And I think that they have to be allowed to have a voice. Otherwise, we're truly gaslighting them in a profound way. And whether or not what they're voicing is true vaccine related, or they're just telling their stories, I know from my personal travels, and you've learned something about my own personal travels that lead me to the person I am now. You know, when I left the Salk, I had post-traumatic stress disorder. I still do. PTSD doesn't leave you. And there are still things that can trigger me. So I live with this. But being able to tell my story a few times has really helped. It's really important for people to be able to share their story with other people. It's therapeutic. And I hope we can stop taking that away.
Yeah, taking that away from the people who are experiencing. Oh.
Because there it is.
There we go.
You noticed me taking out my earbuds and putting them in my charger because it's been a long day of podcasts.
Yes,
I just missed what you just said.
I said,
now I'm back.
No, that's fine. And I just want to end, because this is really why I'm doing this, is to give the people who are experiencing adverse reactions a bit of a voice as well. They're not crazy. It's possible that it's connected to the vaccination, and we can hear that we can hear that as human beings. That doesn't need to be misinformation. That can just be information, and then we can decide for ourselves what actions we want to take in our lives.
Yeah. Thank you for that. As speaking as a long hauler and as a former vaccine recipient, yeah. Okay.
Thank you so much, Dr. Malone. I appreciate this. I'll work on this in the next few days, and I'll touch base. I'll send you an email. I'm actually curious if you could send me any photos of you at the Salk Center, or anything from when are they on your website maybe?
I've got many journalists ask this, and we've got a standard group of photos that we send out, and I'll be glad to send you some.
Thank you so much.
Okay.
Have a fantastic day.
All you got to do is tell me, do you want pictures of horses and me also, or just the Salk?
Yes, send it all.
Okay, you want the whole thing. Okay, good enough. And be good and be brilliant. And thank you for the opportunity to share this time.
Thank you. And I hope I can do this justice, because it's very important.
Okay, thank you.
See you.
Be good. Bye-bye.